Road, and I'm very sorry that this is my first visit to National College. So thank you so much for inviting me, and it's a pleasure to be here and to speak at this historical college that we all have heard about so much. So what I want to do today is introduce to you the human brain and to tell you how historically we have proceeded in understanding about the human brain, and how fascinating it is as a subject, and especially for the students who are here. I also want to say how neuroscience as a discipline bridges across other disciplines, from computer engineering to mathematics, physics, biology and psychology, how it integrates all these different disciplines to help understand the human brain. Could I switch off this slide? So I always start with this quote from Hippocrates, who said about 2000 years ago, when there was a big discussion about what was the seat of our emotions. People believed it was the heart. At that time, Hippocrates wrote, "From the brain and from the brain only arise our pleasures, joys, laughter, pain, grief and tears." So it is the brain that mediates all our senses, makes us the human being we are, creates the culture that we are all aware of. But the brain is not a mirror. It doesn't replicate the outside world like a mirror. It actually, as we look at a picture, if I look at Einstein there, Gandhi there, the emotions that arise in me is very different from what it would arise in the young girl sitting in front of me. So the brain is actually, it recreates, abstracts and recreates and restructures reality, which is a very interesting thing. Because even in vision, you can have no two people who see the same thing or understand it, even though it's the same picture in front of us. So how does the brain do all this? So while the sensory inputs come from the outside world, whether it is as sensory inputs to the eyes, the ears, or the smell, these are brought into the brain, and then the brain plans and executes the response. And the more higher order of the function it is, the more difficult it is for the brain to put this all together. This is enough. This is enough.
Not for you. It is directly going to YouTube.
Okay, yeah. No, this is okay. Oh, you want some photo? Is 84 billion neurons. We are born with these neurons. When we are born, we have the whole gamut of 84 billion neurons. Yeah, it's not as if we make them as we grow along. And there are 100 trillion connections, which together will make up to about 3.2 million kilometers of wire. That's what is the most astounding feature. And all of this in a mass of 1.5 kgs. But then, this represents roughly about what? 1% to 2% of the body weight, but it consumes 20% of the cardiac output of blood, because glucose and oxygen from the blood are essential for the functioning of the brain. And the brain, actually, if you look at it, especially even the brain from a rat or mouse, which is what is the model we work on in the laboratory a lot of times, it's like a jigsaw puzzle as you open it, because it's made up of all discrete nuclei, each of it with a specific function, and then they talk to each other and thus communicate. It's, even as you look at the brain, you're astounded and amazed at the complexity it can produce. So neuroscience is a very young discipline. When I speak in front of physicists, I have to say, when you talk of a couple of 100 years of history of physics, we have a history that dates back about 40 years, over 40 years, and it emerged as an independent discipline from the fringes of psychology at one end of the spectrum, biology and computational science at the other end of the spectrum. So if you look at what is the grand challenge in neuroscience, the grand challenge in neuroscience is to understand the basis of which the information processing occurs at the brain, at the cellular level, at networks and systems, across lifespan, from birth to death, and from normal to diseases. Because in this lecture I have also tried to knit in the burden of brain disorders and how little we know about it and how much it affects our society. I've also done it because historically we have learned more from the diseased or injured brain than from normal brain, over time, and that's how neuroscience has grown and our understanding. So today, a neuroscientist studies the brain across different levels of organization, from molecules. So even as I speak to you and you are trying to remember the number of neurons in the brain is 84 billion, that's you're putting it into long-term memory. So that even if you are asked that question, say maybe 2 years later, you will still remember. If I do that process today now, you're actually synthesizing proteins in the brain, and there's new gene expression that's taking place to help you consolidate this memory and put it into long-term memory. So molecular processes occur even as we try to remember, as we try to recall what we have done. And at the cellular level, changes takes place. The neuron is very dynamic. I've been talking to you about these dendrites and spines that constantly change as we remember, as we learn new things. And then these networks are formed between neurons, which is what helps information processing. And the network is dynamic. It is not at all static. Every minute it changes. And that's the beauty of the brain, ever-changing dynamic object. And actually, it reflects today's society, which is one of the fast-changing societies. All and across the system, say the visual system that I'll be talking to you about from the eye to the back of the brain, and all of this together produces ultimately the behavior, the joy, the sorrow, the pain, the grief. While we study across these different levels of organization, very intricately linked into this is computational science and informatics science. Why does this happen? Because today, in order to understand complex biological systems, the need for the rigor of mathematics and physics cannot be overemphasized. We need to build models, which is what mathematics and physics helps us do, and it is from these models that we can gather more knowledge from the information we generate from our experimental data. So I'm very proud to say that as I started this department at University of Science about 5 years ago, we are now 10 faculty members, and there's only one biologist in that. I have 2 electrical engineers from IITs, 1 aerospace engineer, a physicist. I myself never did biology after my middle school. I'm trained as a chemist, and I turned into a neuroscientist. We have a psychologist, we have a psychiatrist. This should tell you that you are not limited by discipline boundaries in today's world. You are only limited by your own willingness to dream and execute your dreams. So nothing is out of bounds for you. And I want the young people here to actually think of it and not be limited as we were. I know the horrible transition it was for me as a chemist to move into biology, which happened pretty accidentally. But I don't think that should be a hindrance at all in today's world. And I very proudly recruit one more engineer or one more photonics person as they come with ideas of how they can help solve the mystery of the brain. I always start my talks with Hippocrates. I first pay my homage to him. My second homage is to this man, Santiago Ramon y Cajal, whom we acknowledge as the father of modern neuroscience. Cajal was a Spanish anatomist, and he actually had a crude microscope with which he sat in his kitchen and saw these beautiful pictures of neurons in his microscope and drew them from sections of human brain. Santiago Ramon y Cajal was the first person to show that neurons, these single cells, which look very pretty out here, are the ones that actually are the single information processing unit of the brain. He won the Nobel in 1906 along with Golgi, who actually discovered this method to stain the neurons using silver staining. So Cajal had this amazing ability over 100 years ago with very, I should say, crude tools to look at two-dimensional structures and predict how the functional consequences of that. For example, he looked at these structures and was able to show that, actually predict that neurons will process information in a unipolar way. That means there will be one part which will actually receive information, and there will be a single axon that will transmit that information. a lot of what Cahal predicted have all come true in 100 years. He predicted the role of the supportive cells in the brain called glia and their very important role. My other hero, I always feel that today's students don't appreciate history of science as much as they should, because it's the history of discoveries that actually motivate us even today, right? And we need role models, after all, to emulate. And so you will see that throughout this talk, I will keep referring to major scientists who made remarkable contributions. Actually, if you take the Nobels awarded in medical physiology, the maximum number of Nobels have gone to neuroscience. In fact, even last year, the Nobels that went to Strothman and Sudoff were again for neuroscience, or for Kendall, or for Carlson, or for Greengard. You name it, the most important discoveries in neuroscience have always been rewarded by Nobel Prize. So Sherrington was an Englishman who in 1932 got the Nobel for, of course he got the Nobel for looking at visuomotor control, but he coined this word synapse. And today that's a very important word. So synapse is the point where two neurons communicate and talk to each other. One sends information, the other receives information. So the synapse will always have a presynaptic terminal that is transmitting the information that's here, and a postsynaptic terminal that will receive the information. And I just wanted to share with you this picture. I'm sorry, the resolution seems not to be as good as it's on my computer screen. So this just came out last month in Science magazine. So if you look at the synapse, which is this part, so what they have done is they have taken serial sections and done electron microscopy, and this is a reconstruction. So this is the end of the nerve cell. What I showed you in the previous slide, this part, the presynaptic terminal. It has its own mitochondria. So you have the energy production part of the cell present at the synapse to provide. All the neurotransmitters that are needed to transmit information are present in these vesicles. This is the active zone where the neurotransmitter release takes place, and it has all the components of the cell except the nucleus. There is mitochondria, there is endoplasmic reticulum, it has machinery to make protein and also to degrade the protein. And it is made up of about 300,000 proteins. While we have only 23,000 genes, the brain generates diversity by the mechanism of alternate splicing. And this is how it, if you take a cut through the synapse, this is how it looks. And you can see these vesicles sitting there. And these are about 60 proteins that one can see in this kind of a little shape. And if now you look at the postsynaptic, because you need to know about both, the postsynaptic dendrites, these are the spines which receive information. So you can see here in this electron microscope, this is the presynapse with all the vesicles with neurotransmitters. This is the postsynapse that receives the information. And this is what you see as little dots studded along the neurites of a nerve cell. And my colleague Deepak Nair, who is a physicist from IIT Madras, who just joined us, actually uses a technique called super resolution microscopy, where he can look at single molecules of the neurotransmitter receptors in the spine and show how they are studded along the spine with the scaffolding protein to hold the neurotransmitter receptors together. The kind of technology that's available to us now is truly awesome. To be able to visualize one molecule, each single molecule at a synapse in a live cell, a cell that's living, as neurotransmission takes place, was something we never ever thought was possible. So these are things that truly make you, it's very amazing to see this kind of thing. So this is one of my favorite quotes, that between the C. elegans, which is a little worm about a millimeter long, and us, the difference in genes is not much. When the human genome was unraveled, it was a great disappointment to the people who were proponents of the genome, because the number of genes in humans was not tremendously more than in what we see in worms or mouse. Where the difference comes is actually in the number of connections we make. In fact, if you look at a rat brain slice or a human brain slice under a microscope, what astounds you is the complexity of connections in a human brain. Like I like to say, it's the connections that make the difference and not the number of neurons. And those connections keep changing throughout our lives, and I'll tell you about it. So the complexity of the mind is determined by the complexity of the neuronal networks of the nervous system. So I like to stop this. See, Sherrington predicted this. Sherrington was a poet and a great writer. Way back in 1920s, he said, swiftly the brain becomes an enchanted room, where millions of flashing shuttles weave a dissolving pattern. Always a meaningful pattern, though never an abiding one. And in these few words, he described to you what's called the synaptic plasticity. The fact that these networks that we form, they break, they deform. So it's like an enchanted room that keeps on changing. There's always a pattern that's formed, a meaningful pattern, but it's never an abiding pattern because it changes all the time. And that's what I want to tell you about today. This dynamic of the human brain and the enormous potential that it has. So the decade of the 90s were declared as a decade of the brain by the US Congress, and it was adopted by the whole world. And tremendous progress has occurred in the last 25 years from the 90s onwards. And this happened because of two things. At the molecular end, we suddenly had techniques from molecular biology that were not available before. We could clone receptors, we could produce transgenic animals, we could, a new technique called optogenetics came, where we could make neurons fire when we wanted by shining a light. So all these techniques at one end helped us to look at large arrays. But the other end, which made a difference to neuroscience, was the emergence of modern imaging techniques. I know most of you know about MRIs because you see them in every street corner now. But what made a difference was functional MRI, where you look at it's called the gold signal, that is, you look at the oxygen utilization between oxygenated hemoglobin and deoxygenated hemoglobin, which gives you an indication of that part of the brain was active in performing that task. So what fMRI has done is provide a window into the brain while we are awake and we are performing a task. So you lie in the scanner, you're shown a screen, you're asked to remember, you're asked to do mathematics, you're asked to play a game at times, and then your brain is imaged simultaneously to determine how the brain actually functions. I won't talk much about the others, but to go on because I think. So, what I want to tell you today is how does the brain change across lifespan, from gestation to childhood to adolescence, and from adult to aging, which is of concern to a lot of us, including several people in this room. What are the brain disorders that afflict us during the lifespan, and what is the current treatment that's available? So if you look at the disease burden of brain disorders, so disease burden is not what number of people who die of the disease, but the number of people who live with the disease and cause a burden to the society. So that's what is known as disease burden. 1/3 of the disease burden, even in developing countries, is because of brain disorders, which can vary from mental illnesses to childhood disorders, which I've described. There is high morbidity, and there is a lot of stigma. I think even today in our culture, epilepsy and mental illnesses carry a huge stigma. So they are underdiagnosed and undertreated. There are no cures for most of brain disorders. And in this country, in most developing countries, there's a transition taking place. We have eradicated polio. We have eradicated smallpox. So as the shift occurs from infectious diseases to communicable diseases, our burden of diabetes, hypertension, cardiovascular diseases are increasing, whereas the burden of infectious diseases is going down. As this transition occurs, the burden of brain disorders increases. And most importantly, better public health, the vaccination cannot help reduce the burden of brain disorders. And it's a tragedy that our policymakers have not focused on this issue at all. So let me go to where it all starts. Are you able to see it? I think the resolution is poor. Is it poor? It's okay. It's okay? Okay. So, I'm sorry about it. I don't know. So, at 3 weeks of gestation, the brain is the earliest to start forming during gestation and the last to get complete. So in humans, at 3 weeks, we start making neurons at the rate of 250,000 cells per minute at birth. And at birth, we are born with a gamut of 80 billion neurons that we have, and we live with most of our life. But what's the difference between birth and as we develop? The difference is that when we are born, there are very few connections. The neurons are formed, they have migrated to wherever they have, because it's a laid out architectural plan, which is why any kind of childhood inborn errors, that is any genetic disease, the brain is always affected, because one single cue going wrong will change the position of the neuron or the connection it needs to form. But it is experience or the environment that actually builds more connections when the child starts developing from the time of birth. And this outcome is a combination of genes that we inherit and which are expressed, and the environment that the child is exposed to. So I want to give you some examples of this, taking different systems, the visual system, learning, just to give you a sprinkling of how we have learned our understanding of what we do. One of the systems in the brain that has been very well studied has been the visual system. The visual system consists of, from the eye to the back of the brain, which is the primary visual cortex, along with the several what we call supplemental areas. In fact, more than 70% of the cortex is used for visual system, to see and to recognize those objects. Again, I want to tell you about Hubel and Wiesel. Hubel and Wiesel were two scientists who worked together at Harvard and won the Nobel Prize in 1981 for the remarkable contributions they made to visual systems. They were the first to show that the neurons in our back of the brain, visual cortex, react to certain angles of light. So there is a neuron that will only react to this vertical beam of light, and here David Hubel and Dr. Wiesel are trying to understand how much of a visual space that single neuron responds to. But what you will see here as this video goes on is that it will only respond. The noise you are getting is the neuron firing, because that's how the electrical activity is converted to an audio distinct. And you will see that this is the receptive field that one neuron is able to feel. But you see, it's only looking at the light in this direction. And now you will see, as they change the light, that neuron will not respond. So that means this is known as orientation selectivity, which is, there are neurons that will only respond to horizontal or vertical bars of light. This is how the visual system is set up. So what Hubel and Wiesel did was then ask the question, is the visual system completely developed? What will happen if you shut the eyelid in a newborn monkey for 6 months? So what they did, they sutured one eye in a newborn monkey and opened the eye after 6 months. And what did they find? They found that the brain is mostly cross-wired. So the right eye always is wired to the left brain. All of it, we still don't know why that crossfire occurs. So in any case, in the deprived eye, across it there is absolutely no activity. That means the first 6 months that the eye was shut has impacted on the activity of the brain. Right? So then they went ahead and did a series of experiments where they sutured the eye at different time points after birth. At 1 month, still it was deprived. At between 1 and 2 months, it was deprived. But when they did it much later, at 4 months of age, there was no deprivation. Which goes to show that visual connections are formed from the outside world. That is, stimulus from the outside world is what helps us form the visual connection. Which is why the child getting adequate sensory input soon after birth is very, very important for all these systems to develop. So Hubel and Wiese showed how environment is what determines the connections and not genes alone. And my colleague Arun, who is actually an electrical engineer from IIT Bombay, goes further in this to try and see, okay, we know the vision, any object that is there, the vision comes to the back of the head, but how do we recognize that object as a cat or a dog or a bottle? Correct? That's a very important question. That's not something the visual cortex does, but it is this part of the brain. This is the visual cortex. So it's actually this part of the brain, what we call inferior temporal area, which actually is involved in recognizing objects. Now watch this one. So here Arun works with monkeys. He's showing these pictures to monkeys. This neuron is only responding to animals. See that? So there is a neuron in IT which is only responsible for recognizing animals. There's another one that will do recognize only other kinds of objects. But the other important thing is, how does this neuron know? You see here, there are two views of the animals. And the neuron is quickly able to recognize both the views of the animals as the same. And it activates. And that is the beauty of it. That in that fraction of a millisecond, that the view changes from this to this, it is still able to recognize it as the same animal and activates it as similar. So Arun actually works on, that's why he's an electrical engineer, he actually works on how does this coding occur? How is it able to recognize these two views as a single view? Which is where the integration of computation, mathematics and neuroscience all comes together. And I hope you understand now why we have such a diverse set of people. So I just want to switch gears here and talk to you a little about learning. Learning is one of the aspects of human behavior, I think, which is at its highest in humans. It's also there in animals. And we continuously have the ability to acquire new things throughout our life. Maybe a little slow at older age, but we never stop it. But the brain constantly changes as we learn new things. So I want to give you an example, again from the work of one of my colleagues at NBRC, Vanessa. So she uses these birds who learn to sing from their father. And that note that they learn to sing, they have to learn between the age of 20 days to about 30 days. If at this time they don't learn to sing from their father, they never learn the song. So is it with humans. So in humans, from birth to about 12 years is when there's auditory learning, sensory motor integration, and reliance on auditory experience. It's why if a child has a defect in hearing, you want to get it treated before the age of 5. That's very important because the critical period is lost. Afterwards, whatever intervention you do, the brain is hardwired and it won't change. So look at this song. I've just put I hope you can hear it. Yeah, you can. So this is the normal bird song. And then, if a bird that did not hear any sound till about 40 days and then the father taught it, that bird, it never learns to sing the way the father teaches. So the critical periods are very important, and I think it's a great lesson for us, especially the way we teach our children, in the way science is taught in schools. In colleges, by then, it's all mostly hardwired. But I think it's very important to tailor our education to the way the brain develops. And that's what is being done in a lot of countries, such as Japan. So, but what Soumya, who's an anatomist, incidentally, she's from Mysore. So what Soumya showed is that as the bird learns, these are the neural connections, you actually prune off the connections, and by the time the full-fledged song emerges, the brain has pulled off, it's pruned off those connections, cut off those connections that's not needed, and strengthened those that it needs to sing the song. So the brain works by a very simple formula. You use it or you lose it. And I think that's the best lesson that I would expect you to take with you as you leave this hall after the lecture. The more we use the brain, the better we get at it. The less we use it, we just lose it. And it's so obvious. As a child, the few languages we know, if we don't use it as we grow as adults, we never learn them again. I mean, I used to speak Bengali and I can't speak it at all now because I never used it. So it's very important that we continue to remain active, continue to learn new skills regardless of our age to ensure that our brain is active and that it's constantly engaged. Another example is another favorite of mine. I always tell this. Another example of the plasticity is this story of London taxi drivers. I have said this in several of my talks and I hate to give a talk without telling the story. It's such a beautiful story. So Firth et al., Firth and Firth, husband and wife team from London, took a bunch of taxi drivers who drive taxis in London and did structural imaging, MRI imaging on them. So London taxi drivers cannot get a license till they have the map of London in their head. So their spatial memory has to be remarkable. I definitely would not have passed the London taxi driver test. So when they looked at, when they did the MRI, and then they calculated the volume of different parts of the brain involved in memory, hippocampus, they found that the posterior hippocampus volume actually increased, while there was a decrease in their anterior hippocampal volume, right? But what they found, which was even more interesting, is the increase in volume was directly proportional to the number of years the driver drove the taxi. So it clearly shows that the more you engage in an activity, the brain actually changes. We still don't know what this volume increase represents in the brain because there's no way to know. Imaging doesn't tell us that. But look at it. If you engage your brain in a spatial memory task, you're actually changing it, permanently structured. That's how dynamic the brain is. So it also tells you the potential that's there in us as humans to achieve remarkable things by directing our brain to those kind of activities and learning skills. So just to briefly tell you, because I've talked about childhood now and development, look at the kind of burden that these diseases have. Cerebral palsy affects 2 to 3 per 3,000 births. It's unchanged for several decades now. Learning disability and dyslexia affects about 10% of the population, and I think that's a big problem in schools today. One of the things that concern me tremendously in this country is maternal child nutritional deficiency, and I'll tell about it. Because if a mother is anemic, successive generations, even if there is adequate intervention, face cognitive disability. In a country like India, you cannot just imagine the consequence of this burden. Mental retardation and autism, actually autism has increased from 1 in 150 to 1 in 88 in about 6 years around the world. Of course, it's more prevalent in Europe. Coming back to the nutrition deficiency, you know, our politicians and everybody, the policy makers, are very, always talk about this demographic dividend. The demographic dividend is the fact that we have the world's largest working population in the age of 15 to 59 years. In fact, India is going to provide the workforce for the whole world, and we consider it as our demographic dividend because this workforce will earn money for the country. But this dividend will become a disaster if that population is not adequately engaged and is not cognitively competent. And nobody is thinking about this fact that unless maternal nutrition is improved, unless our mothers are well nourished and healthy, we are not going to have cognitively competent people, regardless of what schools we have or what colleges we have. And it's a tragedy of our country that more than 50% of our women are anemic, regardless of the social class. So this is something that I hope somebody here will take notice of and tell about it. Now switching back to coming back to life and the brain, adolescence is the time, a very important time in one's life. Adolescence is a time where we actually see that there is a lot of mood swings. Actually, for a college like yours, it would definitely be seen in the +2 level and maybe even in early undergrad career level. What happens is at this time, a second wave of growth and pruning occurs. See, development is a time when connections are made and cut off very quickly as we learn. In adolescence, the last to have the pruning and develop is the prefrontal cortex. The prefrontal cortex is the part of the brain that makes us the humans we are. It tells us, it's the one that helps us to decide, to make judgment, and to plan actions, being aware of the consequence. Like we will not cross a road when the car is coming because we know if we do, the car will hit us. But in adolescence, unfortunately, this maturity hasn't occurred as yet. While the emotional brain, which is the amygdala, is mature. So their emotions control their actions and not judgment controls their actions. Which is why that's a time when they don't listen, they want to do very dangerous, risky behavior, and it's also the time that they go into addiction issues. Because they don't understand the consequences of getting addicted to drugs. And why that happens is the surge of pleasure that you and me feel when we say run a race, and then the joy that you see in Messi's face as he scores a goal. I mean, that unmistakable joy, uncontrolled joy. Or the joy when people like me feel when our students do well. That joy is, the joy happens because of the release of dopamine in the structure of the brain called the ventral tegmental area, deep inside the brain. The drugs of abuse actually create an artificial surge of dopamine without having done anything. And the whole brain, that we love that surge of dopamine, that feeling of exhilaration, that our whole brain goes towards finding out the next dose of the drug that will give us the dopamine surge. And because in adolescence we are not well, our prefrontal cortex does not act, we tend to go into. Actually, when you are angry, I always tell this to my faculty, so when you are angry, your prefrontal cortex shuts down and your amygdala takes over because your emotion of anger takes over. And you do things that you always often regret. Which is why I tell my colleagues, even when you're angry, don't send that email. Write that email, put it in your draft folder, send it when you're calm, and you can actually judge whether what you wrote is correct. So anger is a time when your judgment goes completely haywire. So it's better at that time never to make a judgment, but to wait till you're calm before you act. So that talked about this. I want to briefly talk about mental illnesses. I know Satish Chandra is coming here in a couple of days to talk about it. So today, of course, the big change that occurred in the 80s and 90s is we understood that mental illnesses are biological in nature and structural. In fact, two twins, identical twins, who have schizophrenia, one shows a dilated ventricle and loss of gray matter, the other doesn't show. And most of these are caused because of imbalance of chemicals in the brain or imbalance of networks. They occur in the prime of life, they are underdiagnosed, undertreated. Lifetime prevalence is 22%. Lifetime prevalence means those who have the disease, those who will have the disease, and those who have had the disease. Now, if you look at years lived with disability, the top contributors are again mental illnesses. I say this because I'm so pained that adequate attention is not given from our public health policies to treatment of mental illnesses. I just want to quickly go into memory and just give you a short brief and take you on to aging. So, memory we have in three forms in temporal way. Short-term memory is what we store in temporary capacity. Most often, it might be the car number of the taxi that I would take to go to a meeting. I would need that number because I know when I have to get out, I have to reach out to that car. But you will forget it immediately. Using of the short-term memory is the working memory. Long-term memory is when you actually, like I remember my son's phone number, because it's so important. So, short-term memory involves modifications of existing proteins, mostly phosphorylation. But long-term memory involves activation of gene expression, new synthesis of proteins, and formation of new connections. And if you look at the way we learn and we memorize and we recall, it differs as we age, although we do the same tasks. So the brain is constantly changing the way it uses its networks for the same task in the same person as we age. So if you look at young and old, to ask them to look at a picture, the young activates a lot of the brain, old much less. But the best part is this, when they are asked to make a decision on the picture, the young people put it in long-term memory, the old don't. So as we age, the short-term memory does decrease, but our ability to consider and make judgment actually increases as we age. Now coming to aging, because that's my area of research, I want to end with that. This is one of the big problems that countries like India will face, because as lifespan is increasing, the number of people who are living above the age of 60 is going to increase. Currently it's over 100 million in India, and you have diseases such as Alzheimer's and Parkinson's that do affect people, and a host of other age-related disorders. Currently there are no treatments. There is absolutely no help that's available. So I've listed a few major diseases here: Huntington's, Parkinson's, Alzheimer's, and motor neuron disease. They affect selective regions of the brain and cause selective dysfunction. For example, Parkinson's disease is a motor dysfunction. But today, as people are living longer with Parkinson's disease, that's the beauty of modern medicine, because we can live longer with the disease. Now, you are now seeing that they are having cognitive deficits also. They are having behavioral dysfunction. Alzheimer's is a disease of the memory and cognition. Motor neuron disease is a terrible disease that affects the neuromuscular junctions, and it's almost like 5 to 7 years after diagnosis the patient dies. I just want to briefly tell you a little bit about Alzheimer's and close my talk. So Alzheimer's, this is Alzheimer's, who described this disease in a lady who was 51-year-old who had rapidly increasing memory impairment. And when he looked at the brain, he saw these huge plaques sitting in the brain. The brain is littered, it's like a dirty floor, and the neurons are all dystrophic. You have dystrophic neurons. And in this situation, if you look at our country, by 2050, we are going to have lots of old people out. So Alzheimer's disease and dementia is going to become a big problem in the developing world. And in fact, WHO predicts that the increase in the numbers, the blue is developed world, red is developing world. We are going to have the largest increase of dementia in the country. And the country is absolutely ill-prepared for this. With nuclear families, no support systems, no proper care for old people. There's no geriatric care of any consequence in this country. So what the current understanding is, because by the time the patient comes, it's too late. He has lost 80% of the neurons. In Parkinson's, 80% of the neurons in that region are gone. So we are not thinking of cures now, but what we are thinking is, if we diagnose early enough the disease, can we slow down the progression so that as the patient lives longer with the disease, he has a better quality of life. So all of the effort in treatment of Alzheimer's is going in that direction, what we call disease-modifying therapies. So I just want to give you a snippet of my own work in my lab. So I am very inspired by Dr. Warrior, an Ayurvedic physician of great repute from Kottakkal, Kerala. And Warrior and me have been having a dialogue for several years, and Warrior has always said, why can't we use the knowledge of Ayurveda for getting better treatments for diseases which have no cure in the allopathic system of medicine? So he gave me several plants, and one of these plants, which is actually ashwagandha, turned out to be amazing in terms of reversing Alzheimer's disease in a mouse model. So these are the kind of models we work. So we take the genes that are known to cause disease, and those genes are actually inserted into the mouse, in germline fashion. So the mouse that are born show the same features of Alzheimer's disease. They're littered with plants, like I said, and they show pathology and behavior. Now I want to show you a small video. So what we do is we took 2 sets of animals. So this is not treated with the extract, what we call vehicle. So whatever we dissolve the extract and we put it. And we have a test to test the mouse on a memory task. So these have have something to eat, and the mouse should go into these, eat and come off. And you will see this mouse, this is how an Alzheimer mouse behaves. It never learns. It's never be able to find its way, what we call this 8-arm radial maze. You see he's kind of stuck there. He just sits there and he's pretty clueless. And now soon you will see the mouse that has taken the extract. See, he's going into all the wrong ones. He should go into 2, 4, 6, 8. He's not going. It's almost like somebody who loses the way, which you typically see in people with Alzheimer's dementia. And now, so quite often in Alzheimer's, so now you see the smart mouse, same, they are brother-sister, or brother-brother I should say. You look here, he will go, he'll eat all the 4 in no time and walk off. Doesn't make a mistake. We just trained him for a week. He's gone to the right one. And you also see the briskness with which this animal that we gave the extract, and he finishes the task in a few seconds, and there's one more left, he'll go do it. They shouldn't enter the one they have gone. They have to find the one they have not gone. And we see that. And what we also found was the extract completely removed all the plaques in the brain. And the interesting thing, I won't go into the details, was this extract acted not on the brain, but actually in the liver. It increased a protein in the liver which was able to pull out this A-beta, which is a causative factor in Alzheimer's, out. So the purified extract that I got from the plant that Varya suggested actually acted in a manner that Ayurveda propounds in terms of saying that the GI functions, the gastrointestinal functions and liver functions are important, and it's body health, not brain health. So this is probably a first evidence that you can treat a brain disorder by targeting not the brain, but actually the liver. So I want to leave you with the thought that as of now, we've probably scratched the surface of understanding the brain. And the grand challenges that are there ahead of us is, how does the human brain work and produce mental activity? Does physical activity in the brain lead to thought, emotion, or other behaviors? Nature or nurture, the genes or the environment? How does the interplay of genes and experience shape our brains? Most importantly, how do we keep our brain healthy? How do we protect and restore our brain as we age? In fact, I'm very happy to tell you that this aspect of the aging brain has been now interested 2 large donors that we have got. Mr. Chris Gopalakrishnan, the Infosys co-founder, has endowed 225 crores, actually 250 crores to ISE to set up a center for brain research that will focus on the aging brain. And what we hope to do is actually to be able to combine clinical research, basic research and large data computing into this. So look at imaging, look at behavior, look at genetics, use at the center and do the kind of things that we've never been able to do. It's a very generous endowment from Mr. Gopalakrishnan. And just 3 months ago, Ratan Tata came to us and gave us another 75 crores to the Center of Neuroscience to study pre-symptomatic Alzheimer's disease. That is, how can we find out if a person is going to go into dementia much before the symptoms are there? And all of us at the Center for Neuroscience, our faculty, we are all involved in this endeavor. So this is a very unique thing in this country that over 300 crores have just suddenly poured in a span of actually last 6 months to focus on the problem of the aging brain. I think that's a very remarkable change and we're all very excited and hopefully Indian Institute of Science will be able to contribute to this. Thank you very much.